Hello everyone. In the previous video we were discussing about how the germ layers are developed from the zygote, which is a part of embryology, and in this video I will be discussing about how the digestive tract develops. Okay, so digestive tract develops after the formation of the germ layers, and on the basis of the development of the digestive tract, some animals are called protostomic animals and some animals are called deuterostomic animals. So we will learn about that, but not directly. I will not give you the definition directly. First we will see how the digestive tract develops as a supplement, okay, and then we will understand what is protostomic and deuterostomic animals, right? So let's get started. In the previous video we had discussed about the formation of germ layers, and here you have seen that we had formed a bilaminar disc over here, which we had to focus on the bilaminar disc, which we had taken out, and then we had learned how this bilaminar disc is growing, right? So actually the bilaminar disc is inside this structure, right? Inside this structure. So to understand the structure completely, we need to put the bilaminar disc inside this structure and move on to understand the later stages. So to do that, I will pause the video and put this structure, roughly drawn structure, over here, over this bilaminar disc, as replacing this bilaminar disc, and then we will move on to see other foldings. And also I have got a nice picture which explains the same process, and it is not as roughly drawn, it is good picture, and I have got it from Wikipedia. And in the last section of the video we will sum up all the things using that picture, which will give you the complete idea. So let's get started. So here I color coded the structure a little bit, but you can see the general portion, that is this portion, this portion, inside portion. This is the embryo portion that we are discussing about. The bilaminar disc developed into this thing. As you can see, this is quite similar to this one. Okay, so I color coded the entire thing, and it will help us to understand the things. So in the previous video I had also discussed that this outer layer of trophoblast cells had to differentiate into syncytial trophoblast cells and cytotrophoblast cells. Okay, so this outer layer will differentiate, and this picture represents that also. That is this portion. These are syncytial trophoblast cells, so they embed in the help to embed the embryo into the endometrium, right? So this is the syncytial trophoblast cells. So let's forget about the cells. I think there is a Y in that syncytial. Okay, so syncytial trophoblast cells, and over here this one, the inner one, are called the cytotrophoblast cells, and they actually remain with the embryo. They do not help in putting the embryo into the endometrium. So this is the cytotrophoblast cells, cytotrophoblast cells. So cells I am not writing anywhere, so that's cytotrophoblast cells. Alright, another thing here, just to show all the things, this is the amniotic cavity over here. Amniotic cavity. Okay, and over here we have the blastocele. This is the blastocele, which will develop into the yolk sac, as we will see very soon. This blastocele is going to develop into the yolk sac. Blastocele. And then the color coded area, that is the bilaminar disc, which has the epiblast layer, and at this point it is also called the trilaminar disc, because you can see 3 layers. Although not 3, there are many layers of the mesoderm, but they are considered as 1 layer only. So there are 3 layers. This is the which will later develop into the ectoderm. So let me write ectoderm only. Okay, so it is in the stage of conversion between the epiblast cells and the ectoderm. So we can write anything, I guess. So ectoderm, mesoderm, and the innermost, or the, here you can see, is the endoderm. It's not completely become the endoderm, neither they have completely become the ectoderm and mesoderm, but I am just writing for this. So endo. So what next? Next we will see how this next stage is called the mesoderm spreading. So what will happen in the mesoderm spreading is that the mesoderm cells are starting to divide and divide and divide, and they become so much that they start to occupy these areas. Okay, they start to form another layer across this blastocele. Okay, so they spread and become across, form another layer across the blastocele. They form another layer across the amniotic cavity over here. Okay, and they also form another layer across the whole structure, that is from here to here. So like that they form, the cells divide so rapidly and they grow and spread around throughout the embryo and form these layers. Okay, so they form the layers, one layer around the, so let me draw it again better. So the mesoderm forms one layer around the blastocele, here you go, and one layer around the amniotic cavity, and it is actually from outside. Okay, so it is including those cells of the cytotrophoblast, and it also forms one layer around this whole structure. Okay, so there's mesoderm spreads around. We didn't discuss about this in the previous video because our focus was not on that, but I thought to discuss the whole thing as I am discussing embryology. So there you go, it forms the around this layer, and this forms the structure. This is around the day 18 of the embryo development, considering zygote to form at day 1. This is at day 18, and the mesoderm spreads like this. Now due to this mesoderm spreading, we have got some more things over here to label. Firstly, we have got this cavity over here. Okay, let us choose black color. This cavity over here, this cavity is called the exocoelom. This is the exocoelom, exocele cavity. Like we wrote blastocele, we can write exocele also. So let us write exocoelom. Okay, so that's a cavity outside our body. Okay, so here the structure will be developing inside the placenta, and then we also have two more labeling, like this layer of mesoderm cells around this whole structure forms the chorion layer of the embryo. So let me make it a little bit thicker so that it becomes easier to understand. Okay, so this is forming a layer, chorion layer, more thicker. Let me choose brush. Okay, with the maximum size, this will do it. So this is forming another layer around the embryo, as you can see. The mesoderm splitting only, and this is called the chorion layer. You may have heard the name, but this is how it develops. Right? This is the chorion layer. Let me label now. This is the chorion. You have heard chorion, amnion, these layers around the embryo. So this is the chorion layer. Let us write chorion only, and the region where it encloses the amniotic cavity over here, don't think I have the enough space over here. The region where it encloses the amniotic cavity over here, this is the amnion layer, right? This is the amnion, chorion and amnion layer. Amniotic cavity encloses means is enclosed inside the amnion layer, which is common sense. The names are not hard to remember. And finally we get the development of the blastocele. Finally we get the blastocele developing into the yolk sac. So this, after this is enclosed over here, this structure is not known as the blastocele now. This is called the yolk sac. Okay, so the yolk sac, we will discuss what it does a little bit in this video only. So this is the yolk sac. Yolk sac. I wrote in red color. It is difficult to maintain these colors over here. Yolk sac. Alright. So these are the structures that have been formed, and now what will happen is that this amniotic cavity over here will expand a little bit and form a structure which I will draw after pausing this video. So let us see. So here at last it was difficult to show all the cells over there one by one. So as we know, we can represent those cells by lines. So you can see these red cells over here, these red areas over here represent all the mesoderm cells, and these outside black area represent the outer syncytial trophoblast cells. This blue area represents those cells, and this yellow represents those yellow cells which were the the exoderm, and this brown cells the endoderm. So I have replaced the cells, and this is how the diagram is like in most books. So very few books show the cells inside it. Alright, so now what will happen is that this exoderm layer is just developing, and it will increase in curvature, and this thing curves even more and then forms the structure as we will see right now. So you can see it forms a structure like this. Okay, so the previous structure I kept it over here. This is like this, and this amniotic cavity expands over here, and it narrows the point of contact between the yolk sac, and the structure becomes like this. See, the point of contact between the yolk sac has been narrowed because the amniotic cavity has been expanded. Okay, and the structure becomes like this, and here comes the formation of the digestive tract, where it is, while the amniotic cavity is closing over here, this mesoderm area, there occurs a gap in this mesodermal area, there occurs a pore in the mesodermal area, and this pore may develop into the anal pore or the mouth, right? So this pore, based on if it develops into the anal pore, then it is called the deuterostomic animals. So this is the main concern of this video, so let us write it over here. So deuterostomic animal, increase it a little bit, anal pore develops first, and in protostomic animals, we get the mouth or the oral pore. Anyways, mouth develops first. Here we go. So you can see the name only suggests this thing. There's not, the terms are not very fancy over here. The proto, we have always seen that proto means early or before, and stoma means mouth. So protostomic means mouth develops first. And deuterostomic means, deutero means after, and stoma means mouth, as we see. So mouth develops after, that means the anal pore develops first. And this this structure, this pore continues inside and then forms the digestive tract of the body of the organism. Okay, so that was the development of the digestive tract, and I hope I gave the understanding of the clear understanding of protostomic and deuterostomic animals. And now, as I promised, let us see on a well drawn picture which I got from Wikipedia on this early embryology, okay, or the embryogenesis. So you can see over here, this is the first zygote with this gray color, that is the zona pellucida, and these are the polar bodies which have developed due to the meiosis of the ovum cell, egg cell. Anyways, that zygote causes divides itself, and this process is called cleavage process, and it forms the morular stage. Okay, so morular stage, there are all undifferentiated cells. Okay, and this is the polar body. This is the second polar body is actually present within the zona pellucida, but it will disintegrate, so it is not a lot of concern about it. And now the thing is that this will form the compaction. So what is compaction? Compaction is actually development of the tight junctions or junctions between the cells, so that when this, and then the differentiation, that is the trophoblast cells are differentiating on the outside and the inner cell mass is there, and due to this compaction, that is the formation of these junctions between the cell, you can see all the cells are attached together, stick together in this place. Or else what would have happened is that one cell would have been floating over here, one cell would have been floating over here, one cell would have been floating over here in the coelomic blastocele. Okay, but that is not happening. Why? Because of that compaction and the formation of junctions over here. Alright, then this differentiation, then cavitation, because the cavity is getting formed, and then the step is the zona hatching. Zona hatching means just a fancy name for that disintegration of the zona pellucida layer. You can see this gray layer is hatching off, okay, like an eggshell. It is hatching, and we are getting this, and the structure will now grow. And at this time, I told in the previous video, at this time the implantation will occur. The trophoblast cells will help in the implantation, and then the inner cell mass will differentiate into the hypoblast layer of cells over here in brown, and the epiblast layer above it, and then the bilaminar disc will form. Alright, and then you can see that this primitive streak has happened over here. It has not shown the longitudinal view, I have shown it, but the primitive streak has been shown over here. Just a point on the primitive streak has been shown, and from the primitive streak you can see the cells are migrating. Some are ending up in between the layers over here, red cells, and some also replace the cells of the hypoblast layer. Okay, now these are the last two stages which I discussed in this video. So the mesoderm will be spreading. So this mesoderm over here, this red color you can see, so this red color mesoderm you can see over here will be spreading all around. Okay, so it will be spreading all around. So let us see how. You can see this red color has spread all around, so the cells have spread all around. It has spread throughout the whole structure as well as it has covered the Yolk sac and the amniotic cavity. And this the cells which have spread all around are called forms the chorion layer, and the cells which cover the amniotic cavity forms the amnion layer. Alright, and the point where the path through where the cells reach the outside of the structure forms the umbilical cord, as you can see over here. This path from where the cells from in the middle of the structure reaches the outside of the structure forms the umbilical cord, and it is also mesodermal cells. Okay, and you can see last amniotic cell and sac enlargement portion. Okay, so amniotic sac enlargement means this amniotic sac is very small right over here, and it has enlarged a bit, and then it has thinned the contact between the Yolk sac and the embryo, and over here the digestive tract will develop. So let me increase the size. So over here the digestive tract will develop, and this digestive tract will grow and then form, and this wherever the digestive tract is developing, this can form the anal pore or the mouth pore or the oral pore. Okay, and that based on that we call protostomic or deuterostomic animals. Until now this is the formation of the digestive tract, then we will have new relation in that embryology only, but this we will not discuss, we will discuss in later some video. But our concern is to understand protostomic and deuterostomic animals, and also to get a good understanding on how the germ layers are developing. Okay, so that was met in these two videos, and in the next video we will continue discussing the characteristics of animals, right? So hope you liked the video. If you liked it, please give it a thumbs up below, and if you disliked it, you can give it a dislike also, but please comment down and subscribe. Bye.